The broadcast is now starting. All attendees are in listen-only mode.
Hello to everyone online. We're just going to wait a couple more seconds to let people join the webinar, and then we will get started. Okay, it is 1 past, so we can get started now. Hi, everyone. On behalf of the Northwest Territories Council on Invasive Species, Pests, and Pathogens, I'd like to thank you all for joining us for the third in a series of webinars to celebrate National Invasive Species Action Week. Today, we are so excited to have a talk on whirling disease by Clayton James. So as a bit of an introduction, Clayton is currently employed as a senior biologist with Fisheries and Oceans Canada, but spent the better part of 3 years from 2017 to 2020 studying whirling disease and its distribution and severity throughout Alberta as a member of Alberta Environment and Parks. More recently, he was contracted by the government of BC to help inform their surveillance efforts for whirling disease in British Columbia, where the parasite currently remains undetected but could present a serious risk. Fish parasites and pathogens are a particular passion of Clayton's and have been since his graduate studies in 2006, where he studied a brain-encysting trematode of fathead minnows at University of Lethbridge. In his spare time, Clayton enjoys watching the Oilers, unfortunately, as well as fishing and spending time with his dog Jasper. So thank you so much for joining us today, Clayton, and I will pass things over for you to start your presentation.
Awesome. Thank you so much for the introduction, Jonah. And thanks again for giving me the opportunity to present some of the work that I previously did in my position with the province of Alberta. And I appreciate everybody's time who's interested in learning about the topic. So the research that I'm going to present today, it's a little bit outdated now. It was done in 2019, but I still consider whirling disease a very emerging threat to specifically Alberta. But I do know of some more current research that's going on in the province, and I'll give you a bit of an update of what I know to date. So just out of curiosity, Jonah, people can see the screen? Everything's good?
Yes, everything looks great.
Awesome. Okay. So what I want to do today is I kind of want to give you guys the story of whirling disease, particularly in Canada. But in order to do that, I'm going to give everybody a life cycle refresher of the parasite responsible for whirling disease. And then I'm going to talk a little bit about the history and the timeline of whirling disease in Alberta, as I know it from my time with the province. And then I'm going to shift focus after that, and I'm going to talk about a very specific study that was conducted on the Crowsnest River, which is a very important fishery in southern Alberta, some work we did in 2019. And as I just mentioned, I'm going to talk a little bit about some of the current research that was actually based off of what we did in 2019 that's still ongoing. So as mentioned, I want to give you an overview of the life cycle that causes whirling disease. And this is my disclaimer that this is really just a slide for illustrative purposes. It is not to scale nor morphologically accurate. But I find it actually a really good slide for visualizing the life cycle. The parasite itself, Myxobolus cerebralis, it requires 2 hosts to complete its complicated life cycle. Everybody tends to think about the fish because that's what's important to us. A lot of folks, fish for recreation, and we do know that whirling disease is a problem for fish. But the other host that doesn't get as much attention, but in my opinion, is much more important for the success of the parasite as well as the severity that you would see in fish, is the tube effects worm. This is a small aquatic worm that lives in the substrates of water bodies. In order to get from one host to the other, the parasite requires 2 spore stages. And the first spore stage I'm going to talk about is the myxospore, and the myxospore comes from the fish host. This is a very hardy spore, so when you talk about transferring invasive species and transferring the disease on your equipment, This is the one that you'd want to look out for because it can last up to about a year in the environment and is kind of resistant to certain treatments. The worm will unknowingly be moving around in the dirt and will feed on these parasites, and then a period of development will occur inside the worm, and then literally the worm is going to poop out the next spore stage, which is called the actinospore or triactinomyxon, and for simplicity we refer to it as a TAM for short. So that's this stage here. So this is like a free floating. It's a very fragile stage. It's only going to last about 6 to 10 days in the water column. Again, this one's not much of a concern for passing the parasite from one water body to another. But this is what infects the fish. And so it'll attach to the skin of the fish host. It will migrate through the nervous system. And what it's seeking out is cartilage tissue, because that's what the parasite feeds on. So one important distinction that I would like to make, and this is essentially the core of my talk, is that there is a big difference between the parasite Myxobolus cerebralis and whirling disease. Often the two terms get mixed up, and that can sometimes confuse information. And why I need to make that distinction is that there are a lot of fish populations out there that have the parasite, but they don't actually have whirling disease. So if a fish gets infected, it's not necessarily going to get the disease and die. There are many watersheds, as we know it in the States, that have had the parasite for decades and have never seen losses as a result of whirling disease. However, that being said, much like all diseases, if under the right circumstances, a fish gets, like if there's environmental circumstances that promote the parasite, then you can see fish get infected badly enough so that they contract the disease. And how you will see the disease is they start to exhibit some of what we call clinical signs. And so this can include a whole range of signs like skeletal deformities and like with the head or the spine. And we see a dark, distinct black tail, and sometimes fish will start doing whirling behavior, which is actually where the disease derives its name. And the reason I have these deformities is because, as I mentioned before, the parasite likes to feed on cartilage. And young fish from about 0 to 9 weeks old, well, they're 100% cartilage. And so if they get infected badly during that time frame, the parasite will do a lot of damage to that cartilage. And then when it becomes bone, it will lead to these deformities. By the time a fish is 10 weeks old, it's completely or mostly ossified, so mostly bone. And fish that get infected after that time generally won't get the disease because there's not enough available cartilage to make a severe impact. Most commonly, rainbow trout, cutthroat trout, brook trout, and mountain whitefish are known to be the most susceptible fish species. I can talk a little bit more about this, and I'm sure folks will have questions about different species that we can talk about a little bit later. I will say that if a young fish gets the parasite and it's severe enough to cause whirling disease and for the fish to exhibit those clinical signs, the odds of it surviving are very low. Another thing to point out, and this is something that I've had many conversations with anglers before, is that if you see a larger fish, which you're angling for, and you see it whirling around, or it has some features like a bent spine, sometimes people will say like, oh my God, this fish has whirling disease. Odds are that's not true. It's probably from some other malady and not actually whirling disease, because as I mentioned, the fish that you're going to see whirling disease for they're going to be like 5 centimeters long. And they usually don't live because if they're whirling around at that stage, they're not able to avoid predators, and they're probably not able to feed themselves as effectively. Once they have them infected and they die, they're going to obviously go down to the substrate. They're going to be fed on by a variety of things, including those worms that are breaking them down, and then those worms are going to ingest the myxospores and then start the life cycle again. So in terms of a story, like where did whirling disease come from? Where did it all start? If you want to go way, way back, you'd have to go back as far as Germany. I didn't even know the date, but it was the parasite originated in Germany and co-evolved with brown trout. The first time that it was observed in North America was actually in Pennsylvania in the 1950s, I think. But it really hit wild populations hard in the mid-90s, and particularly in Colorado and Montana. This figure here I find is an exceptionally powerful figure about the timeline of the parasite's development in the upper Colorado River, which saw 90% plus declines of the rainbow trout population. So what I have here in this figure is the length frequency distribution of rainbow trout captured over time. And so on the z-axis here, you have 1985 essentially down to about 2009. And the x-axis is the length. So these are all different lengths of fish from 10 centimeters to 54. And then on the y-axis, you have your number of fish captured, essentially. So as you can see, preceding whirling disease in the 80s, you have multiple age classes that indicate a healthy population. And those persisted for many years up until about the mid-90s. And mid-90s, you start to see like absolutely nothing of the smaller lengths. And so going back to what he said about the smaller fish being more susceptible and dying, it wipes them out completely. And so what you see is a population with remaining adults available, but no young fish coming in to replace those adults. And so over time, around 2001, these adults are eventually going to die of natural causes of old age, and then you're going to get a complete wipeout. So they saw major, major declines in the Colorado River. So for Alberta, and I guess Canada, because as far as we know, it's only in Alberta, to look at the timeline of the parasite in Alberta, we have to look back at when we actually last tested for it. And this is, again, really important. You can't just go out and say, oh, I saw a fish with whirling disease, so it's been here for 40 years. You actually have to use a molecular assay to confirm the parasite. And that has to be submitted to the Canadian Food Inspection Agency, as they're the jurisdiction that's responsible for them. And so in response to what was happening in the States, in Colorado and Montana, and we all know Montana is quite close to Alberta, there was raised a lot of flags. And so in 1997, a not-for-profit organization with Alberta, as well as the province, conducted very wide surveillance of the province, which included the Red Deer drainage, the Bow River drainage, and the Old Man River drainage, which we know to date all have the parasite. Between 97 and 2001, they didn't see a single positive detection. And then the last known study looking for the parasite was conducted out of Lethbridge Community College, and it was a sentinel cage study. And what I mean by sentinel cage study is that they put fish in a cage that were uninfected, and then they tested them after several months to see if they're infected. And they did that in the Bow River and the Crowsnest River and didn't find any positives. And then really after that point, people just stopped looking. And it wasn't until 2016 in which the first known positive detection of the parasite occurred in Johnson Lake in Banff National Park. This raised a huge alarm, and the province basically undertook an emergency effort to sample all throughout the province in areas where they know vulnerable trout species are. So that's the gray area you see on this map, looking to see the distribution of the parasite. And so that's basically the figure here, the map I'm showing. Anything that's dark red, that's positive for sure using a qPCR molecular assay. The dark green is a negative for sure, and these light green, light red, there was still some uncertain results. But what's really important here, and a takeaway message, is that the likelihood that given previous testing finished in 2003 didn't show anything, and that we did for sure have a positive detection in 2006, suggests that the parasite entered the province at some point between this time frame. And that will refute the common misconception that I've heard from many people, that they believe parasite's been around for a long time. I joined the provincial government in 2017. They formed the Whirling Disease Program. And one of the first things we did was basically we wanted to fill the data gaps. We wanted to know where else in the province the parasite might be. So we contracted out a very large program to sample areas that hadn't been tested or confirmed yet. And so that's represented by the blue in the figure. So about 2018, we started getting, the question from a lot of the public and from some of our provincial biologists, like, okay, great, like the parasite is found in many different watersheds in the province, but like, what's it doing? Is it actually cause for concern? And so at that year, we decided to shift our focus to sampling some watersheds that we knew were positive for the parasite, because a very wise person once said to me, like, an absence of evidence, in this case, whirling disease impact doesn't mean that there's evidence of absence. So we went to a variety of sites that we suspected might be exhibiting the disease because we saw high levels of fish that tested positive for the parasite. And what we found in one of the sites, which is the Crowsnest River, was very alarming. Our field staff observed several young of the year, so hatched this year, rainbow trout and mountain whitefish that were showing clinical signs. As you can see here, there's a bent tail. This rainbow trout here has a really dark, distinct black tail and a shortened operculum here. And so it sort of raised some flags. We're like, oh, these look like these fish could have whirling disease. And another very interesting thing about this is that no matter how much effort we put in, we could only catch fish that hatched this year. So that's represented right here in this 2018 figure. Anything below this line is hatched this year. Anything above this line would be years 1 and 2 year old fish. And as you can see, we had great difficulty capturing those. If you compare that to 2000, in which the similar method was used to collect fish in a similar area, they had no issue catching 1 and 2 year olds. So this kind of reminds me a little bit of that figure in Colorado. So that's, although, we did the electrofishing and we saw the clinical signs, we couldn't say definitively it was whirling disease. And so we decided to take 2019, do a very comprehensive study to examine the severity of the parasite on the Crowsnest River. And in order to be very, very sure of what we were seeing, we wanted to examine every single part of the parasite's life cycle. So we looked at a variety of metrics. Essentially, we looked at the parasite in the water column. We looked at how the fish could survive and the prevalence of the parasite in the fish. We looked at myxospores by doing some histopathology, and we looked at the worm for the density. And we also looked at environmental conditions, like their optimal temperature regime, which I'm going to talk a little bit more about. First, though, I'm going to give you an idea of sort of what we did for site selection. We selected 6 sites on the Crowsnest River. So this is the upper river, so the flow direction is this way. So we have one at the very top of the watershed. This is the headwaters of Crowsnest Lake, and then several sites going down all the way to almost to the Old Man Reservoir. We also selected a site on the Old Man River because the purpose of this was to serve as sort of a negative control, because previous testing, we had never detected the parasite in this section of the Old Man River. However, in previous testing on the lower Crowsnest River, you can see these red dots that they indicate where we detected the parasite. There were none in the upper watershed, but there wasn't a lot of testing at that point. So at each one of these sites, we installed sentinel cages, and with the intent of eventually housing fish that were uninfected. So we knew they were uninfected. And this is would allow us to understand sort of the distribution of the parasite throughout the watershed, but as well as the severity of the parasite. This is an example of one of the cages that we designed. And right here, you can see there's a mesh screen on the front, there's a mesh screen on the back. That's to allow for the flow through of water. And we also attached a PVC pipe around it for flotation. And the purpose for this was that as water levels changed, the cage would simply adjust with the water levels. On top of that, we added a very rigid top to keep prying eyes out, as well as wildlife out. So this is an example of what these sites, what these cages look like in the fields. This is the top of the watershed going downstream, basically anchored in somewhere near the tallow egg and allowing water to flow through. This is called about the middle of the watershed and down further lower, and then one on the negative control in the Old Man River. So I previously mentioned we did all sorts of metrics and how those can correlate to infection levels. So let's go through those. The first thing we wanted to do was we wanted to monitor temperature at each site, because the parasite responsible for whirling disease, there's a specific thermal window which really speeds up their development and their release, and that's from about 10 to 17 degrees Celsius. So we installed hobo loggers at each one of the sites. Second thing we did is we wanted to examine worm density and abundance, because in my opinion, that is the biggest driver of disease. The more worms you have, the more parasites you have released, and the more severe the parasite will be in fish. On top of that, we wanted to correlate that and actually look at the water for the free-floating TAM stage, doing this filtration technique, which I'll describe a little bit more in the next few slides here. So basically what we're actually doing is looking for the physical parasite. And then the other thing was looking at fish. So we would put fish in these cages that were uninfected, and then we would test them later. One of the things we looked at was prevalence, and the other was how well they survived in the cages. There's two phases to that study. So as you can see, we were quite busy, but we wanted to again make sure that we had a weight of evidence approach to be confident in our conclusion. But we weren't alone. We didn't reinvent the wheel here. We actually looked down to the US, to some experts that have been working with whirling disease a lot longer than us. In particular, we had the help of a gentleman, a retired research scientist out of Colorado Division of Parks and Wildlife. His name is Barry Naring. If you ever search literature on whirling disease, he comes up the most. And so basically, he was kind enough to come visit our cruise and teach us some of these techniques, particularly that filtration technique, which he spent a lot of time validating. For the filtration technique, I guess just simply to give you a simple overview of what we do, you pour 2,000 liters of water through this very, very fine filter, and then you get this resulting retentate, and you basically bring that to the lab under a microscope. So just to remind everybody, this is the stage that we're looking for. It's that free-floating stage, the TAM. Yeah, so we bring it to the lab, we stain them up, and look through the microscope for them. This here is an example of what they look like up close. So this is a TAM. This was one of the first TAMs that we found on the Crowsnest River. We essentially did those filtrations from June to October over the season, I think weekly. So in Barry Naring's vast experience, he basically stated, if you could find one single TAM in one sampling event over 12 different sampling events, so only one, even if you went out 12 times, that's sufficient to cause significant population level declines in wild self-sustaining rainbow trout populations. And keep that in mind. To shift focus a little bit, I mentioned that we did also some electrofishing at each site, because we wanted to look at the wild fish again to see if what we saw in 2018 was consistent. We did all the sites except for this uppermost site because there was no defined channel. And the idea was to look for signs of the disease like we saw in 2018. To give you an idea of what this means from a field perspective, I have a couple of photos here. I actually captured both of these fish at the same site. The one on the left here is representative of a healthy young of the year rainbow trout, and the one on the right is a good example of a whirling disease impacted young of the year. Stuff that we look for, obviously the black tail really stands out. On top of that, you can see the tail's not straight. It's because there's a deformity in the spine. This is a little bit more subtle to see, but it's quite a bit more obvious when you're looking at the fish. They have a bulging eye. And the reason for the bulging eye is that the nose of the fish, the snout, is greatly reduced. So essentially the parasite is just destroying and eating all that cartilage and shrinking, and it makes the eye look like it's popping out. One of the more common ones that you see is a deterioration of the gill plates. And it's obvious in the sense that you can see here very clearly, you can see the gills of the fish. So the parasite has been eating away at the gill plate, whereas in the healthy individual, you can't see that. This particular example, there's a malformity with the jaw. This would be your typical subverted jaw, and here you can see it's not subverted at all. It's inverted. So I would see this fish, and this would be a good indication that it's heavily impacted by worms. So what did we find? Well, I'll first talk about temperature. This figure here is, I absolutely love this figure. This is an average of 4 sites that we did on the Crowsnest River. And essentially what it's doing is we're looking at the average daily water temperature. And the yellow, or surrounding that, is 1 standard deviation from the mean on either side. As I mentioned before, the parasite thrives in a temperature of 10 to 17. So that's represented by this light gray window. So this is when the temperature is ideal for the parasite. Anything below that is fine. The parasite will still develop. It just takes longer to develop, and probably you won't see as much of an impact of fish. If it gets higher, it's actually really bad for the parasite. At about 20 degrees Celsius, the parasite no longer is released from the worm. And there's evidence from a lab experiments, I say that because that's different than field, but in lab experiments, there's evidence that if the temperature gets up to 125, that worms will actually be purged of the infection. So that's very interesting. We have temperatures ranging from about March to almost November. This dark gray box is what's important. So this dark gray box represents essentially the beginning of hatch of rainbow trout in the system to 9 weeks plus their development. So this is when this dark gray window is when fish are most susceptible. So that young fish, fully cartilage, most susceptible. It's a range because the hatch occurs over a range. So at all of our sites, looking at this, anytime this temperature overlaps, you're saying essentially it's perfect temperatures for the parasite. And this is the top of our watershed at CR6. So during that maximum rainbow trout susceptibility, 96% of the time the temperature's in that window. So that's pretty scary stuff. Or I'll just say ideal temperatures. And essentially in the Crowsnest River, it ranged from 76 to 96%. The Old Man River itself, which is our negative control, was 74%, which is also a very large overlapping range. So a result for our worm density, the sites, the values here that are represented are basically worms collected per minute. So you do a kick-neck sample, you put it in a white tray, and then you start picking out worms. So in this sense, you could only find 0.2 worms per minute, or probably, what would my math be on that? You'd find a worm every 5 minutes or so. So very low at the top of the watershed, as well as our negative control. But going down, you can see the numbers get a bit higher, like one worm per minute to almost 3 worms per minute. And then they peak in this lower area, like 10 to 11 worms per minute. So this is what I would consider extremely high density of worms. To give you some perspective, that would take, 2 field staff only 15 minutes to be able to individually pick out 300 worms. So just load it. The second map here result is showing the temps. So how many TAMs over the summer? So we did weekly sampling events, which I think equated to about 14 sampling events. Did we find? Well, we didn't find any at the top of the watershed. We didn't find any in our negative control, but we did find some pretty much throughout the watershed below the headwaters. And as you can see, large numbers of TAMs were collected at CR2 and CR4. So it's, I mean, I'm just going to back up here a slide, but it's interesting. You've got your very high density worms here, and then you got your high density TAMs directly downstream. So there's a very strong link there. And if you recall, I said that in Barry's experience in the studies that he did, if you found one TAM over 12 months, 12 sampling events, that's significant climb. Well, we found 20 over 14, so it's quite high. This is just another way to visualize it. So each one of our sites are represented going down. So this is the negative control site at the top, Old Man River, and then this is the top of the watershed at the Crowsnest River by the Crowsnest Lake, and then we're going downstream progressively. On this axis here, we have TAMs per liter, so not just individual TAMs, but calculated for volume, and then each sampling event. So essentially, any increase from 0 indicates that TAMs were found. The difference between this figure and the figure I presented before is that just because we found TAMs with a microscope, we still had to confirm that they were in fact Myxobolus cerebralis. So we did have to send our samples, like our filtrate samples, to the lab for testing. And basically anything with a red dot was positive for M. cerebralis. Green triangle meant that they didn't come out positive. But we're talking about a very limited amount, like microscopic amounts of DNA here. So it didn't surprise me that you get some examples where we found a TAM, but we didn't get a positive qPCR hit, but then you'd get a positive qPCR hit, but no actual physical TAM found. In terms of statistical significance, this is strongly correlated. So there's no statistical difference between using qPCR or looking under a microscope. And this is really important because I think the way forwards for surveillance is to actually just do qPCR because it's time saving and it's logistically less challenging. Okay. So when we compare our findings in the Crowsnest River based on TAMs per liter, and we compare that to water bodies in the Colorado River where they saw major declines in fish populations, we rank out pretty high, almost like top 5. And for example, that figure I showed you of the Colorado River and the huge decimation population, that's actually had a lower TAM density compared to the Crowsnest River. The ones highlighted pink are specifically where they saw greater than 90% declines in rainbow trout populations. But that doesn't mean the ones that aren't highlighted didn't see huge population crashes. These are like brook trout populations or possibly brown trout populations. This is one of my favorite slides. So this is the first phase of the sentinel cages, so where we put the fish in. And what we are basically looking for is prevalence of the parasite. How quickly do fish become infected? So we put about 100 or so fish in each one of these cages that were reared in the lab. And every week we'd take out about 12 to 15, then we'd send them to the lab to get qPCR'd and tested for the parasite. And then that proportion, we would say that's how many were positive for the parasite. So you got this graph up here that shows 7 days, 14 days, 20, and so forth. And then the percentage of the fish that we took out for that week that were infected. And then we have the numbers here. So this is starting, everyone has 0. We go 1 week in, our upper site's 0, all the way down to the middle, our control site 0. But this is, to me, very incredible. You see this huge spike, 93 to 100% of fish after 1 week in these cages tested positive for the parasite. Move on to week 2, and then you start seeing it's like 100 across the board at the bottom, and it's up the watershed. So the extent of the parasites through the watershed is quite high. Next step, you got even higher, going again, each site is higher. And then week 4, this really weird thing because no study is perfect. All of a sudden we had lower percentages. And the reason I think this occurred was because when we are netting fish, so the parasite does have an impact on a fish's health and then their ability to swim. So I think we were just biasly sampling of fish that were infected in the first few weeks, and then all of a sudden we captured fish that were a little bit harder to catch and were able to maybe avoid the parasite in the cages. But week 5 saw a resurgence in the percentages. Again, it's very, very high. And then week 6 was the highest for everything. So it had up to 52% of the fish in the cage up high in the watershed were infected. And interestingly, we found a positive detection in our negative control, meaning that's not really a great negative control. So this actually served as the basis for determining that the parasite was in this watershed. And if I were looking for the parasite in the future, I would find sentinel cages are a really good way to test for them, because you don't actually have to take fish samples out of the lake or the river. You can just put in aquaculture fish and then test them at the end of the summer. Phase 2 of our fish in the cages study was to actually take gametes from rainbow trout in a tributary of the Crow, hatch them in the lab, and then put them back into the cages and just look at their survival. And it was quite interesting. Not very many fish tended to survive in these cages because they were quite young, and I don't think they have a high survival rate in general. So you saw about 36% at the top of a watershed. This, for whatever reason, CR5 had a high percentage survive, but you can see all down here, this is significantly less fish survived, to as low as 14%. So there was some impact to survival over between August 22nd and October 30th. Really, this all boils down to is the lower watershed. All these results align. It's very strong evidence that the Whirling disease is certainly impacting the lower part of the watershed, and eventually over time will probably move up. To look at some of those electrofishing results of the wild fish, not our cage fish, we wanted to basically look for, what percentage of fish are we capturing that have clinical signs. And yeah, you can see clinical signs were quite low, like only 8% of fish here, 19%, but we had as high as 84% of fish that had clinical signs, some form of clinical signs, whether it's a gill, whether it's a slumping head, whether it's a blacktail, etc. To give you an example of our catch per unit effort, like I said, last year, sorry, not last year, sorry, 2018, we caught some of the freshly hatched fish, but we didn't find any of the fish in the 1 year older range, except for a couple in CR2. Very much the same in 2019. So there's indication that there's still an adult population available to spawn and produce these guys, but they're not living, they're not surviving past that year. Higher up in the watershed, you could see a little bit more survival, but again, that also matches the results that I showed previously. The provincial biologists really didn't believe us as much, so they wanted to go out and they did their own float, complementary float e-fishing study, and were very surprised to learn that yes, in fact, Crowsnest River is not doing well, particularly in the lower watershed. So you can see adults exist pretty much at certain parts in the watershed, so they're still contributing to these younger fish, but these low watershed ones, it was pretty much a ghost town. So again, this supports that there's a major die-off occurring in Crowsnest River. So CR2 flagged out as our highest, everything was the highest, the most worms, the most hams, the most everything. So we did a bit of a pivot in the field, and we decided to set up a second cage at CR2 and just put some of the wild fish in, because we wanted to see what happened to them over time, because they would have a natural infection and would progress similar to what you would see with the local population. And so what we found was what I had mentioned before: almost all fish had severe clinical signs. You see the blacktail, so obvious, the bulging eyes, the shortened operculum. This is a really good example of the bulging eyes. You see how much it looks like his eyes popping out of his head almost. After 2 weeks in the cage, so I think we put 30 in, and after 2 weeks, there was only 11 left. I have a video here. I'm not sure if it's going to work. It never works really well, but hopefully you can see it. So this is a video of what the whirling behavior looks like. So a lot of, it's not a slow moving whirling behavior. It's considered like a rapid tail chasing behavior. And you can see that in one of the fish in this video here. So if you look, focus on the middle of the screen, I'm hoping you see it. I'll play it a couple times. So look at this guy here. Oh, it's really slow. Like you can see on this one here, I don't know how well you guys can see that, but there's a distinct blacktail, but try this again. Essentially this guy right here, yeah, doesn't look like it's working very well, but he does a very rapid tail chase. So when you scare them, their alarm response isn't to move away. Their alarm response when they get scared is they just spin around chasing. It looks like they're chasing their tail. And you can see how that wouldn't be a very effective survival strategy. We did send, so at the end of the study, there was only one fish remaining, and that was this guy here from that wild tank. And we wanted to see what it looked like on the inside, just to confirm even further that whirling disease was having an impact. And he did some histology, and this is an example of a vertebra with normal cartilage cells. And this was actually one of our fish from CR6. So completely normal, and this was the fish from CR2 down in the lower watershed. Essentially, all of those cartilage cells were displaced by myxospores feeding on it. So, very, very heavily infected. Yeah, so that was a summary of the study. We know for a fact that whirling disease is having a major impact in a watershed in southern Alberta. It's unfortunate because the Crowsnest River is a world-renowned fishery. But the takeaway that I wanted to get from this study was that there is a distinct difference between infected with a parasite and having whirling disease. So this is a fish that looks normal, and this is one severely impacted by whirling disease. And we only did the one site. Alberta is a very big province, as you all know, and there are lots of areas where the parasite is. But unfortunately, there was change in government recently. The whirling disease program lost its funding. It was reorganized, and many, if not all of these, have not been looked at any further for actual whirling disease. But that being said, there are still people that are carrying on the torch, we'll say, and using some of the stuff we found from the Crowsnest River to be able to apply it to other areas in the province. I have a very good example here. There is a student out of Lethbridge Community College. His name's Kelly Reel. Really, really awesome young man. He basically took what we had looked at in terms of temperature and that optimal temperature range, and he applied it to an area of the Upper Oldman. So the Upper Oldman is really important because it's a population of federally listed species at risk, the West Slope cutthroat trout, that the province is actually trying to recover these populations. And there's no known positive detection of the parasite in that area. But there was a positive detection downstream in the Oldman River. So it may happen over time. So essentially what Kelly was looking at is like, well, are the temperatures conducive to the disease? And so that's represented by these red percentages here. So this is your time spent during the most vulnerable stage of fish, the time spent between 10 to 17 degrees, which is optimal for the parasite and for disease outbreak. As you can see here, comparative to the Crowsnest River, where we saw values as high as like 96%, there are quite a bit of lower values, 20, 34, even sometimes 0%, where the temperatures don't go. This would tell me that, whirling disease is maybe a little less likely, but it's not impossible because there is a large chunk of overlap. So you can see areas with 60% overlap, 58. The next step, I think, in studying this for Kelly, if he wants to continue to pursue this, would be to check for the worms. Like, are the worms present and that are high abundance? If they're not present and they're not in a high abundance, it's probably not, and odds of a whirling disease outbreak happening are probably not that high. Another colleague of mine, Chloe Christensen, she actually worked with us on the Crowsnest River project. She was one of our field technicians. After her tenure with the province, she decided to do her master's to look for whirling disease using that filtration technique. She wanted to put her emphasis on the Bow River, which is another blue ribbon fishery, world-renowned fishery. And the reason why she wanted to put her emphasis there is there's a lot of recent literature coming out that's saying, hey, you know, like rainbow trout populations are in major decline. So the province has been looking at this for years, and they're seeing large drops. This is a CBC article showing 50% drop. And while the article does, you know, does mention that, you know, there's multiple, they call it multiple smoking gun stressors. They say it could be flooding, it could be whirling disease, but generally the mind frame or the mindset for the province is that it's actually caused by anglers and catch and release mortality. In my opinion, being that I like studying fish disease, I think, you know, it's important that we look at those smoking guns, and really Chloe is the only person trained besides myself that could go and look for that signs of disease. So her and some fellow students, they did filtrations on multiple sites of the Upper Bow, similar to what we did in the Crowsnest River. And what she found was not really surprising to me. Is anything that's not yellow shows that they found towns and that disease is likely occurring. So if we pull out the positive sites, these are the watersheds that are likely in the Upper Bow, and these are all tributaries feeding the Bow, including the Bow main stem. They're all likely experiencing at least some level of whirling disease. I remember I mentioned it's 1 in 12 sampling events to get a positive. Well, she only did 5 sampling events, but to get like multiple positives is a very concerning thing. Particularly these 2 sites, Jumping Pound and Sebald, they are actually known West Low cutthroat trout watersheds. So that's kind of scary. For her, I think next step would be to actually go and do some electrofishing and see if you can find what we found in the Crowsnest River in terms of signs of the disease. So yeah, that would be hers. So I see I only have about 13 minutes, which I think is a good amount of time for questions. That is a summary of the study that we conducted on the Crowsnest River, and sort of what current research is heading towards. So with that, happy to answer any questions that you may have.
Perfect. Thank you so much, Clayton, for that great presentation. Super interesting and very useful, I think. I will invite attendees, if you have any questions, you can put them in the chat. You should have on your control panel an option to open up a chat. You can put your question in there, and I will read it out to Clayton. I'm not seeing any immediately, so I am going to jump in with my own question. I have a few, but one of them is looking at these results and being, I mean, they're pretty scary to look at, and obviously this disease is having an impact and it is spreading in these watersheds. So I'm wondering what researchers and, you know, government and people who are doing work out in the field, what kind of like decontamination protocols are they using for their gear, or like what can they do to help prevent the spread?
Yeah, you know, it's a really good question. When I was with the Whirling disease program, we had a specific group that specialized with decontamination, and they built a pretty rigorous protocol for decontamination, and it was built on essentially the risk that you could spread the parasite to watershed. So we'd identify watersheds that are positive, so that would be your highest risk. So you'd want to do a certain level of decontamination, and that would include like chemical decontamination. Quaternary compound is it's just called quat, is the #1 compound that is good at disinfecting your gear. But in areas where it was lower risk, say that maybe there aren't rainbow trout present, or aren't susceptible species present, there's been no signs of the parasite, generally you'd go down to a lower level, which is like the clean, drain, dry. And provincially, it was if you were doing any work in these watersheds, it was your legal responsibility to follow that decontamination protocol. But unfortunately, like the same protocol doesn't apply to members of the public. And so there's only encouragement to do clean, dry, dry. And I know there's a lot of work that happens in these rivers, but I know there's a lot more fishing that happens in these rivers. And so there is a high possibility of spread through recreational traffic. They'd had a wonderful educational campaign, and that's really what you have to do is you have to educate and because a lot of people that like fishing on the Bow River, they want to preserve the Bow River. So they are willing to do the extra stewardship actions necessary to keep the parasite from spreading. And it's not everywhere. We can prevent it from getting into sensitive habitats or areas where it isn't already. I think initially we responded by putting signs up to say like whirling disease at risk so that people on the ground could see the signs. But I think that actually was a problem because that diluted the message. It basically looked like the whirling disease parasite was everywhere. So people thought, oh, there's nothing I can do about it because it's already everywhere. So if I were to inform a newer jurisdiction that doesn't have the parasite, I would say if you do get the parasite, make note, put signs up where the parasite is, but not everywhere where it could be. If that makes sense.
Yeah, that makes sense. And that's a good idea. I'm still not seeing questions in the chat. Hopefully folks are finding the chat if they're able or if they're wanting to ask a question. I think you can also raise your hand if you want to be taken off mute and ask your question verbally. But anyways, seeing none, I have some more questions. One of them, I feel like the answer might be kind of obvious, but I'll ask it anyway. Because I'm assuming that there are, there's hatcheries or aquaculture in Alberta, and I was wondering if whirling disease has ever been detected in any of those facilities, but my guess is not, since they probably don't have the secondary host, the worm.
You know what? Your question is actually very astute, and this is actually a very, very important factor in the spread of whirling disease. Yeah, so I have a bit of conspiracy theory that I'm going to share with you here. It's just my opinion. It's not based on fact. Okay. But in all of the major jurisdictions in the U.S. that saw impacts to wild populations, it started with hatcheries. And there are a lot of what we call earthen bottom pond hatcheries. So they aren't sealed. So they did have the worm host, and they wouldn't be able to get rid of it unless they did a full decontamination and changed the substrate of their hatcheries. And in the States, they stock their rivers. So that really made it bad. In fact, they knowingly stocked whirling disease infected fish. Like they knew they were infected and still stock them. And so I think the spread in the States was expedited because of their hatchery practices. In Alberta in 2016, there's public hatcheries, and they're well maintained, they're decontaminated. There was never anything that tested positive. But 5 of our private facilities tested positive for the parasite. Because nobody really regulates that. Like there are regulations for their importing and exporting, but I don't know how hard they look. And so they tested positive. A couple of them were also earthen bottom. In fact, one hatchery was directly connected to the Elbow River, which feeds the Bow. And it's really hard to say, like, did the hatchery give the parasite to the Bow, or did the Bow give the parasite to the hatchery? It's hard to say one way or another. But my conspiracy theory is that, yeah, I believe that the reason why we have the parasite is from hatcheries.
Oh my goodness.
And what I think happened is because we did a trace out for those 5 infected hatcheries, the federal government did a trace out, and they connected those hatcheries to 380 water bodies that were stocked from those hatcheries that tested positive. And there's a lot of water bodies in and around the Bow River, like small trout ponds that were stocked by those hatcheries. And so my theory is that the flood event in 2013, if there was fish stocked in those, the flood event, basically those ponds got flooded out, went back into the Bow, like everything became the Bow River during that flood. And I think that really, again, exacerbated it, like spread the parasite really quickly.
Wow.
And that's why you see it. It's prevalent in the Bow. It's everywhere in the Bow.
Yeah.
So.
Oh, wow.
That's just a theory, though.
Yeah. Well, it's an interesting theory. We have a question in the chat. Somebody asked, has any population been assessed for resistance after multiple generations of exposure?
Yes. Yeah, that's a really good question. I could just go on and on about this. So it takes about, according to Barry and other experts that we've discussed, 30 years. So you get the parasite, you undergo the major declines. If you have surviving fish that make it through the genetic resistance, it is inherent and they can pass it on. And they say about 30 years before a population gains resistance and starts recovering. A good example of that is the Madison River in Montana. They're starting to see rainbow trout populations come back. They didn't do anything. They just let them get destroyed. However, they stocked and they still stock. Alberta's not in that situation, right? Because we have cumulative effects. If whirling disease knocks it out, we already know that things like forestry, habitat fragmentation, all those other angling, all those things are still pressures. We're not stocking these rivers. So who's to say they will come back? But there is evidence that over time, populations will gain a resistance. Colorado, they went super high end on their studies for resistance. They actually bred, there's a rainbow trout known, it's a German rainbow trout known as the Hofer strain. It's completely resistant to the parasite because it co-evolved in Germany with this in the aquaculture facilities. They bred that strain with the Colorado River strain of rainbow trout. And they created what they called like this hybrid and a resistant one. And it was more resistant to the parasite. And so they started stocking it. However, it was a really bad fishing fish, like people didn't like angling for it because it was really like they just weren't fun, I guess. But over time, they repeatedly stocked these resistant fish throughout the state, and now they've discovered a naturalized population in the Gunnison River that's a really high quality angling fish that's resistant to the parasite. And now they're taking that fish, bringing it to their hatcheries, and creating a brood stock, and now we're stocking them up. So if it up to me, which it's not up to me, nothing's up to me. If it were up to me, I would be stocking those fish in the Crowsnest River because the Crowsnest River fish are not native. The rainbow trout in there are not native. They came from Europeans way back when. That's a perfect river for trout. You might as well get ahead of the curve, put some resistant fish in there. There are things to consider. It's connected to pure West Slope cutthroat trout habitat. So there's always going to be chance for hybridization and things like that. So smarter people than me would have to assess that first.
Cool. Oh, that's so fascinating. Well, we just have a couple minutes left and I'm not seeing any more questions. So I think I will wrap up. Thank you again so much, Clayton. This was a really interesting presentation. And also thank you to everyone online who joined us for the webinar today. If you'd like to join us for more great webinars this week, you can find more information at our website. That's nwtkisp.ca. I'll spell that out. It's nwtcispp.ca. We also have our Facebook page. Just search for NWT Council on Invasive Species, Pests, and Pathogens on Facebook. And because it's Invasive Species Action Week, I also want to remind folks that you can find more information on our website about what you can do to help prevent the introduction and spread of invasive species, pests, and pathogens. For example, our Clean Drain Dry program highlights the importance of making sure that you don't accidentally carry aquatic invasive species between water bodies on your watercraft or gear. So there's more information about that on our website and how you can help protect our waters by always making sure to clean, drain, and dry. Yeah, I think that's all. So big thanks again to our presenter, Clayton James. Thank you so much. And thank you to
You bet.
everyone online for taking part. So
yeah, thank you all. Appreciate it.
yeah,
Appreciate the opportunity.
awesome. I will log us all out of the webinar. So take care, everyone.
Yeah.
Thanks again.
Take care. See ya.
Bye.